2018 and GATE in the year 2020. Ms. Srishti got a Sofura Scholarship in 2005 and 2009, Ishan Udyan Scholarship in BIODIVERSITY for the year 2015 to 18, Indian Academy of Sciences Summer Research Fellowship in 2019. With this bright academic career, Srishti Lekha got Kanazura University Grant Type Scholarship in 2020. She is expert in bioinformatics softwares, Autodoc, Cytoscape, Mega6, Pymol, Chimera, etc. Also, she has become expert in gel electrophoresis, PCR cloning, primer designing, immunoblotting techniques, DNA RNA extraction techniques, qPCR, bacterial transformation, preparation of single cell suspension, etc. Today, we are lucky to have Srishti Lekha among us through this virtual platform. Thank you, Srishti, for being with us. And over to you, Sankranti.
Srishti Lekha ma'am, you can start your presentation.
Yeah, thank you very much, Sangeeta for your nice introduction, and also Dr. Jogen Kalita sir for inviting me to this platform. It is a great opportunity for me to get such a good platform to have this, to deliver this talk. Okay, so I'll be sharing my slides. Uh, could you please tell me, is it visible?
Yes, ma'am, it is visible.
Okay, thank you. Okay, so in this talk, we will be mainly focusing on the fundamentals, basics of biomechanics, and then I'll be heading into the organoid mechanics. So before coming into the details, let's have a look at the definition. So we can see that the word itself here says that it is consisting of two parts, that is bio and then mechanics. So bio here is the, we all know bio means life, and mechanics that deals with the physical forces, the action of forces upon any physical body. So the word biomechanics defines the mechanics that is applied to biology. That means to conclude how different mechanical forces can help shaping of an organism if we consider from the biological point of view. This is the main motive of studying biomechanics, and here I'm going to talk about the just a generalized objective, that the main objective is to understand how actually the biological systems sense, transduce, and regulate the responses to the mechanical cues. Maybe they have been that have been sensed from the external environment or from the tissues, within the tissues, from inside the biological system. So I'll be mainly talking about tissue mechanics, one of the very broad category of biomechanics, that it has been a very emerging field, because we all know that our cellular system is under constant dynamicity. That includes motility, differentiation, the gene expression, and we can, or it has been reported that motility, differentiation, gene expression, and many more such dynamic events have some contribution towards eliciting some kind of mechanical responses that again helps in various embryogenetic events, organogenetic events, etc. So today, studies have been provided a biochemical framework to understand such mechanotransaction process and responses of cells to such stimuli. But still, the challenge still remains till date to understand how actually such cells sense mechanical stimuli and then trigger some subsequent biochemical reaction. So this tissue deformation is a very well-known phenomena that we can see in tissue mechanics. So the tissue deformation can occur due to various methods. Like, since tissue deformation is, though it is the word deformation is there, but its importance is very useful in development, tissue homeostasis, wound healing, and various disease condition. If the tissue deformation is again deformed, they may lead to some kind of disease condition. And they have been reported to be elicited by cell division, since we all know that when a single cell gives rise to two cell or four cells, depending on the cell division, then absolutely that single cell got deformed because it has formed now two or four. And similarly, cell shape changes. From here, the picture at the right hand side only, we can see that how the elongated structure got flattened. And this flattened, the process of this is also under, we can include it under tissue deformation. And likewise, cell rearrangement, cell extrusion, they are all dependent or all lead to tissue deformation. Now, the question comes, what actually mediates such deformation in the tissue? For example, when we consider the mechanics acting on a physical body, then we can talk about some kind of stresses. Okay, so the similar kind of stresses can be present or we can observe within a tissue system. They can be tension, compression, shear, bending, torsion, or any kind of combined of any of these. So, such deformations are determined by the mechanical balance. So, for a structure to get deformed, there has to be some kind of balance between the active forces. Active force here defines the force that has been generated inside the cells, and the passive response. Passive response comes from the surrounding cells. So how the forces from inside and from outside are balanced, that actually determines the deformations. And there are some important terminologies used in tissue mechanics. These are constriction, relaxation, stiffness, etc. So constriction and relaxation, they are mainly found to be involved or associated with ectomyosin contractility. We all know that there is an actin filament and myosin motor protein. When the motor protein moves along the actin filament, then there is a kind of contractility that leads to. Okay, so that contractility can be can be defined by these two words in tissue mechanics. These are constriction, relaxation, and stiffness. Stiffness can be, we can talk about the stiffness of the surrounding substrate or the surrounding matrix, because cells, when it grows, it needs some kind of physical attachment to its matrix where it is growing, and also the stiffness of the tissue itself, how stiff the tissue is. These are all comes under the mechanical factors that determines such a deformation. And there are several scopes of biomechanics. So one of the very famous phenomena is spaceflight osteopenia. So when astronauts travels for a long duration in space, they are found to suffer from bone mass loss. Okay, so this loss of bone mass is a similar condition we can compare with the osteoporosis. You must have all heard about osteoporosis, where actually it is prevalent among women, where they are found to have loss of bone mass. So some similar condition was observed in astronauts, and scientists have formulated that it might be due to the microgravity that is present in space, due to the negligible gravitational force, that no pull by the gravity, that means, and the stress that might lead to some kind of aberration of the bone dynamicity, because bone, though it looks like a very stiff tissue, but is very dynamic. Okay, so here only we can see the importance of such external mechanical forces in our physiology, the impact of such mechanics in physiology. So there is this another important study or interesting study is the dynamics of blood vessel. Blood vessel is a very dynamic structure that changes its dynamics by controlling its diameter size. By controlling the diameter size, they actually changes from one state to another, and it is determined by the pressure exerted by the blood flow and a smooth muscle tone that lines the wall of the blood vessel. So the pressure altogether determines the dynamicity. There is this uh, another classification, that is sports biomechanics. Uh, so it is this aims to analyze in a detailed way the sport related movements so as to minimize the risk of injury during a performance and thereby improving the athlete performances. And another field is organoid mechanics. I will be talking about it in the later section. So, what are organoids actually? Organoids are, the word itself says that it contains the word organ if we carefully look at it. So, the organoid are nothing but the mini organ like structures grown in culture, but providing them a 3D space, three dimensional space. When we provide a three dimensional space, cells were able cells are able to recapitulate the structure and functional repertoire of their in vivo counterpart. To some extent, though, not it is possible. It has got some limitations as well, but it is far better than the 2D model that we normally use. Okay, so at the right hand side of the slide, you can see a beautiful picture of fluorescently tagged section. This is actually a section of human brain organoid. Okay, and the different color that you can see in different region defines the regional identity of the cerebral tissue. So I'll talk about a little bit about the difference between 2D and 3D culture because it's very important to know how the organoids are grown to study their related mechanics and stuff because mechanics is possible only in 3D environment and it's very difficult to study in the 2D system. So in 2D culture, the stem cell, I am talking about the stem cell culture. So this is the blastocyst stage of an embryo that contains the inner cell mass surrounded by trophoectoderm. This inner cell mass contains a group of cells, that is the embryonic stem cells. All the biology people will know, this is the basic. So this embryonic stem cell has the property of pluripotency. That means it can give rise to any kind of cell type. So if they are isolated and cultured with appropriate media condition, they can be given rise to any kind of cell types like this, but they are all obtained in 2D. I'm showing here 3D structures, but actually they are obtained in 2D. So here is what happened. What happened in 3D culture? Okay. So similarly, they take the pluripotent stem cells obtained either from embryonic stem cells or induced pluripotent stem cells. Induced pluripotent stem cells, these are obtained from adult stem cells. How they are transformed, reprogramming is done. So when these pluripotent stem cells are again put in different culture differentiation media, accordingly they will form the organoids. And also same with the case of adult stem cell. Organ specific adult stem cells also has this property, but their structural the capability of producing organoids may differ. So I'd also like to introduce you to the culture system. What is happening inside the culture system? Into this culture, We can see here we usually use a tea flask to culture the mammalian cells. Here we get a monolayer. Okay, so if we compare with the 3D cell culture, then we can find not a monolayer but some aggregates of cells. We also call them spheroids. So here in this case, we provide those aggregate a 3D environment to grow. By providing them a 3D environment, we can mimic the actual cell to cell interaction and also cell to matrix interaction in them that actually is going on inside an actual human being or any organism. So we can see here the mesh work with the with the, within the pink. So this mesh works is the extracellular matrix cytoskeletal meshwork that helps also in organogenetic events. And also at the right side, we can see that there is a 2D monolayer and three types of 3D culture. So there are three types. And organoids can be grown at the top of the matrix, and in some cases, they can be grown within the matrix, and at the last part, they can be grown in suspension. So it depends what kind of organoid you are going to develop, because some organ system might need some more stiff extracellular components. So this is just a comparison. Now I'll come to the organoid mechanics. Why to study organoid mechanics? As I have already told, that our organ system is under constant dynamic situation. So to understand those situations, we already know that there are some contributions of mechanics, okay, in organogenesis, but to better understand it, we need organoid mechanics or a clear picture. And also the driving forces behind the tissue deformations going on inside an organoid also helps to define the mechanical forces and how mechanical and biochemical cues are coupled. Because also it is not either mechanical or biochemical, it has to be together. It has to come together to have a better input or an outcome with a functioning organ system. So these all together, we can say that organoid mechanics possesses an important or a promising tool. It can be used as a promising tool to study mechanics. I'll give you here an example of optic cup morphogenesis for better understanding of the tissue mechanics that I have been talking about. So what is an optic cup actually? It is nothing but an eye-forming tissue that after differentiation gives rise to neural retina. I'll use NR and retinal pigment epithelium, RPE. Okay, I'll be using the word INR and RPE in the later parts. So, in the right side of the slide, you can see there is a semi-spherical type tissue that is the neuroepithelial tissue. We all know that retinal tissue, the origin of retinal tissue is neuroectoderm. So when they cultured the neuroepithelial, they first formed an optic vesicle. An optic vesicle, when they undergo differentiation, they are able to form at the middle NR and surrounding RPE. Together they constitute the retinal tissue that are later used to hold the lens that helps us with the vision. And this is the morphogenetic processes that has been carried out years ago. And here the green sheet-like structure is nothing but the neuroepithelial tissue. And in this tissue, I'll give you the polarity of the tissue because it is important to know the polarity when it starts its morphogenetic events. It comes into the picture, and here we can see that the apical surface is the inner wall and the basal is the outer wall. And they have categorized the morphogenetic processes into 4 phases. In phase 1, as I have already told, in phase 1, what happens? The optic vesicle or the near spherical structure gets differentiated into NR and surrounding RPE. In phase 2, the distal portion of NR get a little bit flattened, and they call this phenomena as apical relaxation. Relaxation because that apical surface is relaxed or become less stiff, become more flexible. That means they are now able to deform. Okay, and when they comes to phase 3, or they proceed to phase 3, they found that the distal part is little bent as compared to the phase 1 or 2. And also the boundary cells, the boundary between NR and RP is also able to form an angle flexure like structure, and this was found to be mediated by some constriction. And in phase 4, they have found that the optic cup tissue like structure was obtained by further invagination of the NR tissue. Okay, so it was the general biochemical experiment that outlines the phases of optic cup formation. And in the later years, so some group of other scientists, but from the same lab, has tried recollected all the experimental results obtained in the previous studies and performed some simulation-based study to actually have a look at what is the mechanics going on inside that morphogenetic process. And then they perform some kind of mechanical perturbation methods like laser ablation, microtweezer, strain producing experiment, and use a vertex model, 3D vertex model, to simulate all the predictions. And they found that the boundary folding that I was talking about as the angle flexured part in the previous, in the phase 3 angle flexure part, that is recapitulated in this vertex model as well, and found that boundary cell folding was due to the apical constriction and lateral constriction. Okay, we will talk about what are apical and lateral constriction. And they found that the bending force that is generated at the apical surface, that is denoted by the blue dashed structure that are pointing downwards. I hope you can see it. Okay. So that bending force is somehow propagated to the boundary cells towards the green arrow mark. These cells then were found to constrict. They were found to constrict due to a generation of strain. This is a mechanical strain that was produced at the basal surface. I have shown here in the box below that how the basal surface cells got stretched, or they were under strain, that triggered the lateral lateral constriction that was pointing towards the tissue thickness, that actually was triggering the formation of that hinge structure or the boundary folding that we were talking about in the earlier case. And this hinge structure was facilitating the further invagination of the NR tissue in an apically convex direction. As you can see, I hope you can, I'm sure you can understand the convexity and concavity, where when we see at the blue dashed part, that is the apical portion, that is convex, that is a convex structure. So they named it as apically convex invagination. So, and the whole feedback mechanism, they named it as mechanical feedback mechanism. So, what are the tissue deformations they actually observed here? One is apical constriction at the boundary. Okay, so it was found that those apical constriction at the boundary was due to the elevated myosin accumulation at those apical tissues, apical surface, that made the surface to constrict towards each other, like the arrow is showing, and that ultimately bent the structure in apically concave direction. So this is one of the tissue deformation. There is apical constriction at the boundary and apical relaxation at anor, the middle tissue that we were talking about. So it was found to be due to the reduced myosin activity at the apical surface. This is just the reverse of the apical constriction that was happening due to elevated myosin activity, and here it is happening due to the reduced myosin activity. So of course the phenomena will be reversed. Here the surface is apically convex, the apical constriction was apically concave. And the last tissue deformation they outlined was lateral constriction at the boundary, which was along the apicobasal axis. That's why I talked about the polarity, uh, that that it was towards the tissue thickness, and they called it as lateral constriction. And it was, as I have shown, was found to be due to the strain produced at the basal surface. So there are some other mechanical factors besides the organogenetic processes or the organogenetic mechanics. The effect of matrix stiffness is a very important factor because, as I already told, that it is very important to know where the organ is growing. Is it growing in a very stiff or rigid matrix, or a very loose matrix? So it depends from tissue to tissue. And it has been observed that cell proliferation and morphology varies with varied matrix stiffness. And there is another phenomena called mechano-osmotic regulation. It is a very recent research that shows that intestinal crypt morphogenesis is triggered by the expression of some membrane transporters that was responsible for reducing the lumen volume in those crypt organoids that actually lead to the morphogenetic events in the crypts. So let's have a look at the future outlook of organoid mechanics. As I have already told, even if it recapitulates some parts of the organ system, but it's not very easy, and also there are various associated limitations to recapitulate a full organ system as their in vivo counterpart. So scientists have been trying, although, to enhance the system, and it can be done by engineering the tissues. For example, by incorporating a particular gene, by injecting the gene, we can enhance some kind of biochemical events or even mechanical events that can help enhance the morphogenetic events even better, to better recapitulate the organ system. And also we can manipulate the cell niche, the niche where the cell is growing. Like I have told, the ECM, the extracellular matrix components, we can modulate. And another one is in regenerative medicine. So regenerative medicine, what is done that we all know, when a damaged tissue is there, it can be replaced by some healthier tissue system. And if we can overcome the limitations of organoid, it is possible to replace those damaged tissues with a fully overcome organoid system, a healthy organ system, to recover the unhealthy one. So these are the future outlooks, and this is how the organoid mechanics works. Thank you.
Thank you so much, Kristileka. Yeah, we are proud of you. Very
Thank you,
exciting
sir.
work with the future application. And I am sure the scientists will be really successful in obtaining organs from the laboratory. So your work is very important. Your planning is unique. Therefore, you are getting a scholarships in a country, in a university, top-ranking university in the world, in Japan. So thank you so much, Roberto Songkanti, for comment. And
Thank you,
Songkanti,
sir.
after your comment, please request Hilal Duty Sina to say a few words, then interaction will start for 20 minutes
Sure.
only.
Sure, sir. Sure. So, this was really very interesting work. Actually, from my point of view, I have never seen such type of work in such a means this particular developmental biology prospect in such a beautiful concept I have seen today. So thank you, Shrishtilekha ma'am, for making this possible for us to see the actually see the future prospects of such an important work. I am really blessed actually to see this. So
Thank
thank
you.
you for your beautiful work. Thank you for presenting your beautiful work. And so I would like to now request Professor Hilojyoti Sinha to kindly comment on this. Sir, over to you.
Thank you. First of all, I would like to salute or namaskar my Professor Guru Jagan Chandra Kolita sir. Because of you only I am here. And I'd like also to thank Dr. Shaila Yasmin, my senior in Oregon Muslim University. She has been listening and encouraging the ladies. That is good. So, given the opportunity, I would like to tell one by one. To me, both the lectures are very good and ranged from one dimension to another. One is hardcore field work, another is very much hardcore lab work. So the contrast, that's why congratulations to the organizers to bring the two dimensions together in the same platform to give the audience a distinctive, I mean, katta mitha or kind of crackjack tests. So anyway, so first of all, I would like to congratulate Isa because I had met her in one of the international conferences where she had presented, and I was amused by her presentation actually. And then we had a relationship like a whatever you can say guru shishya kind of thing. All the time we'll keep it, and she has been following all those things. And once I would like to say my heartiest thanks to Sir Jagan Chandra Kolita sir because she, as Shaila Madam has already opined, that he has been instrumental to encouraging the people. So when he came to know about Isa, that she was also taking part in our webinar, then immediately she contacted her through me that okay, would she be available? Would he also? And when he came to know that Shaila Yasmin Appa will be listening to these two young ladies, then immediately he told me that would be she available for some little speech. So both the ways it is very good. Thank you, sir. But Isa did a wonderful job in Malaysia, that too in rainforest. Very, very difficult task. And whoever worked in field, they can understand it. And being a lady, a young lady, that too in Malaysia, in a jungle, everywhere, every danger is there. And she is still, I mean, regularly attending this kind of webinar where there is no electricity, there is no internet connection. Still, she has been working so nice, and she presented in her own way. That means how acoustics can be useful tool to even find a status as well as the availability of some apps which are not otherwise elusive. So thanks and congratulations to Isa. Next, I would come to know about Sri Srilekha. So whatever the biography has been uttered by our organizer Sangeeta Das, I was amazed to see the evolution of the young girl from Tezpur because her education background, she won none other than Sofra Award. So it speaks a volume actually. Many of us may not understand that she has come from a vernacular medium. And now what fluid, I mean, what smoothness she is speaking today. Beautiful English, beautiful pronunciation, every word is understandable, so flawless English, very good. And that proves again the vernacular medium is not a, barrier for learning higher education. And that's why she won all this award. That means she is able to write article from the beginning. And that's why my message again to the young people, not only scientific paper, you must contribute writing some popular article should be understandable by the layman as well. Your contribution to science not only among should be circulating around the scientists, but also to other people as well. Today, Sisti Lekha has already done the job because the way she has delivered the lecture, even laymen like me, for example, I have been working in wildlife ecology for long. and almost I forgot zoology. And I could understand, and it is a new dimension. Else, already Sankranti has pointed it out, the how people can think of a new dimension. That means innovative ideas she has brought in, and that too she has got so much high quality lab facility because of her own ability. Only she could go there, and our blessings are always with her. And hope she will be doing something new for us. And as I just now written, the shell niche is an interesting term for me. I have learned a new terminal term, and whatever she has delivered, I understood and learned a lot, and hope the other audiences also have learned new things, new dimension. And both way, the two lectures are wonderful in their own way. Thank you very much.
Thank you very much,
Thank you so
sir.
much, for your beautiful words. Now we are moving on to the interactive session. So we have our first question for Isa that Himshikhar Kalita has asked, ma'am, are gibbons new world apes or old world apes?
Okay, for the gibbon, they are all world apes because they are restricted to southeastern tropical and also southern subtropical region in Asia. So they are all world apes. Okay, can you hear me?
Thank you. Yeah, yeah.
Thank you, Isa ma'am. So moving on to our next question, we have a question for Sristilekha ma'am by Himshikar Bhuya. He asks, I have a question, ma'am. If organs are produced by stem cell cultivation, do we need to use anti-rejection drugs to suppress the immune system? It is a question for Sristilekha ma'am.
Yes, yes. Um, I too am very new to this field. Please, please, I'd like to say forgive this if there is any mistake. For what I think about this question is that we all know that for when there is a transplantation, there is a graft rejection. That thing is there. So, since these organoids, the stem cell cultivated organoids, are also some kind of foreign, it will be foreign to the system when we transplant it to the actual organism. So, I think that such use of anti-rejection drugs would be should be there. We need to inject such drugs, but I will. But this seems to be a very interesting question. I will also look at it. Thank you. Thank you for that
Thank you,
question.
ma'am. Then moving on to our next question. It is for Iza ma'am. It is asked by Himshikhar Kalita. He asks, ma'am, which natural predators do gibbons have?
For the gibbon, their natural predators are owl, python, and also eagle.
Okay,
Yeah,
ma'am.
yeah.
Thank you, ma'am.
Okay, thank you. Next question we have for Sister Dekhama. Rinpa Lokar has asked how homeostasis is maintained during the organoid development.
Okay. Hmm. Homeostasis, as I talked, like it is the balance between the, if we consider it from the mechanical point of view, then also we can see that there is a balance. There has to be a balance between the forces generated within the cells and also from the forces that we sense from the outside. Okay, so in that way, in from the mechanic point of view, there has to be such maintenance, or the, what to say, the maintenance of the equilibrium, so as to perform such integrity or maintain such integrity of the homeostasis or the cell homeostasis also. But I think there are always some kind of homeostasis going on from the stem cell point of view also, as the homeostasis, like from when there is a damaged tissue, it has to be replaced, right? So when there's has to be some replacement, then there are stem cells that replenish that damage. So there is always going on some biochemical point of view also there are going on homeostasis, and also from mechanical point of view there has to be some kind of homeostasis for the proper maintenance. Thank you.
Sankranti,
Yes, sir.
Sankranti.
Yes, sir.
Yeah, I just want to, okay, inquire from both the promising speakers. The question is the same, simple. So the ethical restrictions for IJA also, IJA Adrena, as well as to Sistilekha, because they are work, both of them, they are very promising worker, and both of them, they are with some sorts of restrictions. Though one in the field, other in the lab, but ethics, the limitations of ethics, both the speakers. So please, you request them to okay say something about ethical restrictions and the way out.
Okay, thank you, sir, for the question. There is an advancement of using 3D organoids over 2D cells, and because 2D cells, when we obtain it from the mammalian cells, it has got some kind of ethical issues. But when we obtain the 3D organoids, the preliminary stages, of course, will having, because the extraction or isolation of the stem cells from, if it is the organoid is produced from a tumor patient, for from the tumor cells, for say, then there has to be some consent. There has to be some consent taken from the patient. But in general, if we want to study the developmental aspects of the organogenesis, for example, then organoid culture has not faced such ethical problems. This is a great advancement to use organoids other than the other cell replacements. So,
Thank you, Sileka. Thank you.
yeah,
So what
thank you.
about Ija now? Ija Adrena, Ija Adriana.
Yeah, thanks, Prof. For the given, actually, we for our study we don't have any ethical issue because we don't take any given sample or any blood sample or anything. We don't take. We just using uh for acoustic survey, which is we just using our ears and listen to the given song and record the data. So no ethical issue. The issue is because of the given is endangered species, and we are working with protected, totally protected area. So we need to ask permission to do the research in protected area, and then also ask the permission if we can do this survey for this endangered species. That's the only thing we have to do. Just ask the permit research, and then we can go for the field work.
Thank you, Ija Adriana. Thank you so much. Really, very hard work. Professor Hilojudi Singh also said that in Malaysia, in terrain, it is not easy. So we are proud of you. Thank you so much.
Thank you, Prof.
Thank you, Isa. Actually, I have a question for Kristilekha ma'am. Ma'am, you have said that the astronauts they go through means they suffer from osteoporosis. So, do they have any means? Do they have any protective something like measurement so as to they do not go through osteoporosis condition?
Uh, Actually, this is actually an emerging field where they can study what are the shortcomings. Okay, when they go to the space, what actually happens in space and what does not in Earth. So they compare And according to the force that they do not experience in space, as I have told you, to the microgravity or the negligible gravitational pull, some stresses are absent or the mechanical pulls forces are absent. So to compensate such absence of forces, they actually prescribe, the physicians prescribe those astronauts to cope up with those environments, some exercises, and also the diet list is very important actually. Because we know the main bone component is calcium, so they basically suggest the calcium rich food content or dietary lists that they provide. So this is what they do, and this is a very advancement, right? Like, to know that. And also, it is not exactly the osteoporosis. I have just compared that. It is a condition similar to osteoporosis. Yeah. Thank you.
Thank you, ma'am. Then we have another question.
Yes.
Ma'am, it is for you. So the question is from Dipanjana. She asks, I have a question that nowadays people are suffering from different types of diseases due to which some of their organs are not working. So that they need organs and also organ donors. And also sometimes due to lack of organ, they die. Is it possible to culture the, is it possible to the whole needed organs, ma'am, and what exact time will it take to grow a full organ? Ma'am,
Yeah, this question
question
seems
from,
similar to what I have talked about by growing
yes, yes.
organoids. So, while growing an organoid, actually, we don't need the problem of donor or the recipient. Okay, we just user friendly, we can grow without knowing the donors because stem cells are already there. It's just, as I have answered to sir also, that it is just during the extraction point that we need the consent and all, and after that the donors. And these are, I think, there has to be some kind of rejection or the compatibility thing is there due to the blood vessel or the vascularity that is maybe present within the organoids. As she is asking, yes, it is possible to culture the whole needed organ, as we have said, using organoids. So we can direct an organoid to form a particular organ type using appropriate condition, culture condition, medium, growth factors, whichever needed, and along with the extracellular matrix. Yeah, it will give into a full organ. Yes. Thank you.
Thank you so much, ma'am.
Okay, thank you, ma'am. We have another question for you only. That question is being asked by Arindam Datta. He has asked that are normal organoids matched to cancer organoids? Also, why do tumor organoids develop from adult stem cells?
Okay. It's an interesting question, actually. I'll try to answer. Okay. I think I'm giving my insights. I am not very confident though. So, what happens during tumor or tumorigenesis is that tumor is already happening in a specified tissue. Okay. That is already committed. It's not in the stem cell form, though all the tumor cells got some kind of stem cells, tumor stem cells that we name it. So these has to be adult stem cells because they are present in the committed tumor cells. So uh, there, he's asking, why do tumor organoids develop from adult stem cells? I think this is the reason because tumor cells are growing inside an adult cell that contains adult stem cells. So we need those adult stem cells to grow the tumoroids. We also call tumor organoids tumoroids. I think he got the answer. This is also again my insights. I hope it is helpful. Thank you.
Thank you. Thank you, ma'am. Next question we have for Riza, ma'am. that Himshikhar Kalita has asked, during which periods of the day are gibbons most active?
Okay, thanks for the question. Uh, the the active period for the gibbon usually in the morning around 7 a.m. to 11 a.m. Yeah, that's around that period the most active gibbon for calling and singing. So,
Okay, thank you. We have another question for you. That what is the main food habits of gibbon? Gibbons are completely arboreal species. Then how can they consume water in the winter season?
sorry, winter.
What is the main food habits of gibbon?
Mm-hmm.
Since gibbons are completely arboreal species, then how can they consume water in the winter season? Hello, ma'am. Can you hear me?
No, sorry, it's lagging. Can you repeat the question?
Yeah,
Okay, sure.
actually,
This question,
Ija, how how they take water because they always there on the top of the trees. So how they take water
Uh,
because they are when you think about their food habit, they need to take water. So question is how they take water because they are always on the top of on the trees.
trees.
yeah. Yeah, thanks. Very interesting question. Yeah, for the gibbon, actually they consume the water from fruit because they totally arboreal. Uh, I mean, they are frugivorous and they consume the water from fruit, and also in the branch, the tree branch actually when the rain come, it will contain the water in the branch, and then they consume from the branch also.
Yeah,
That's the way.
nice answer. Yeah, I also thought of about that, and yes, he is correctly giving the answer. Very nice. Yeah, they are getting enough
This
water
this
from
question was
fruit.
asked by Ranjit Kalita,
Yeah,
Ranjit
yeah, very,
Kakoti.
Thank you, Isa ma'am. We have another question from Bhaskar Ranjan Bora, and it is for Sristilekha ma'am. Is the zero gravity environment of space more suitable for growing organoids and performing other biological experiments?
Yeah, okay. I read that question. I don't know why did he thought that it would be more suitable. I don't think that it will in a space where there is zero gravity. Okay, we have already seen that the effect of low gravity or absence of gravity, what happens to the astronauts. So I don't think that for performing an experiment, uh, space will be a good environment for that, suitable. But if we can, because we all know for a culture to work, it needs all the optimal conditions like pH, temperature is another important factor. If such conditions or optimal conditions are maintained in space, then in space also it is possible. But in general, I don't think that space is a very good place to perform experiments. Okay, thanks.
Thank you so much, ma'am.
Ma'am, we have another question for Shrishtilekha ma'am only. Is it possible to grow any type of organ in the body, or are there any limitations? This question is from Shrishti Das.
Okay. Um, to grow any type of organ in the body. What does he exactly mean? Actually, I didn't get.
I think she means that any type of organs can be grown uh, by this organoid mechanism. I think so. She's asking about that.
Yeah. Oh, so but she's using in the body
Yeah, yeah.
because
I think she might herself clarify what is she asking.
yeah, yeah, because I'll just have a
If
comment. Um, because if there is a damaged organ, then the organ is damaged, right? So if on the top of that we again try to, even if we, it is an ideal answer of course, even if we try to grow that organ in that environment, it might get damaged again, or if it is due to injury, then there might be some possibility. But actually I'm not very clear with the question.
Thank you, ma'am. We have, I think this will be the last question from Himshikhar Kalita for Isa ma'am. Uh, so he is asking that unlike other mammals, why do gibbons differ in size? He means that the female is bit larger than the male.
Yeah, very interesting. But I'm not sure how to answer this question because in my opinion, uh, the male is larger than female of the gibbon. But yeah, maybe because of the sexual body size deformation, so it's maybe have a little bit different between female and male, um, female and male gibbon. I think so. So maybe I'm not sure how to answer. Yes.
Okay, thank you. Uh, Shrishti Lekha ma'am, Shrishti has clarified her question. She means that to replace a damaged organ, the organ will be grown outside. But like, is it possible to grow any organ with this artificial process? Maybe she is asking inside the body.
Okay. um Replace the damaging organ will be grown outside, but like, is it okay? So as I have told, that it might not be possible. I think it is not. Otherwise, there has not to have a very common answer that then such technologies might have not been evolved if it was possible within the body itself. But there are also some kind of reaction, adverse reaction may happen even if it is tried to do so. Because if we try to grow an organ which is not normal, which is not required, but also I am not very clear still that is she talking about growing an organ for the damaged tissue itself within the human or the individual which is undergoing the damage, or she's talking about to grow the organ in some other, not within the body. She has also outside environment, of course, as I have discussed, the organoids are grown outside the body actually. To clarify, I have explained how are they grown in using 3D culture. So after cultivating them in culture, they can be replaced. And also to clarify, this organoid replacements are not yet to, it is not yet used. Okay, so scientists are still trying to develop it into using in regenerative medicine. So this is this only I will say they are trying, but first, they are trying to overcome the limitations that are associated with such organoids that is yet to be recapitulated fully. Yeah. Thank you, Shrishti.
Thank you. Thank you so much, Isa ma'am and Shrishti Lekha ma'am, for your wonderful presentation as well as your spontaneous answers. So, moving on to the next part, we have in our agenda that our dynamic head of the department, Department of Zoology, Guwahati University, and also the General Secretary of ZSA, that is Zoological Society of Assam, Professor Jugen Chandra Kalita, will speak a few words. Over to you, sir. Sir, you are on mute, sir.
Actually, I will declare the closing with my comment. So therefore, you go to vote of thanks
Okay, sir.
now. Yeah.
Okay, sure, sir. Sure, sir. So, as we have come to the end of this beautiful session, I would like to request Ms. Priyanka Kashyap who is a research scholar of Department of Zoology, Guwahati University, to kindly offer the vote of thanks. Over to you, Priyanka.
Am I audible to you?
Yeah, yeah.
Yes.
Yes.
Audible.
A very good afternoon to all the respected members present here today. It was indeed an inspiring and motivating session. I'm Priyanka Kashyap on behalf of Research Scholar Forum, Department of Zoology, Guwahati University, feel privileged to offer the vote of thanks in this International Students Webinar on Fundamentals of Biomechanics and Biodiversity Management. At the very beginning, I would like to express my heartfelt gratitude to our honorable chief guest, come speakers, Ms. Nurul Iza Adrina Binti Mohammad Rameli, PhD scholar, School of Biological Science, University Sains, Malaysia, and Ms. Sristilekha Nath, PhD scholar, Nano Life Science Institute, Kanazawa University, Ishikawa, Japan, for giving their valuable time and gracing today's webinar with their enlightening words on Voice of the Singing Ape in the Rainforest Peninsular Malaysia and organoid mechanics. And I'm sure all of us are motivated from today's session. Looking forward for more of your motivational talks in the future. Thank you for this wonderful session. I'm also very much thankful to our respected invitees. I would like to thank Dr. Shahala Yasmin Ma'am, HOD of Patna Women College, for her gracious presence here today and enlightening today's occasion. She did her PhD from AMU Wildlife Science. She had also qualified JRF NET. I'm also very much thankful to respected Hillal Jyoti Sinha sir from Assam University for being here today and who have always been so encouraging and supportive every time. I would like to express my profound gratitude to the head of the Department of Zoology, Guwahati University, Professor Jogen Chandra Kolita sir, for his constant encouragement and support, and without whom this event would not have been possible. And I'm sure under your guidance, we will achieve great heights in the coming years. Thank you so much, sir. And also, I would like to thank our joint coordinators, Ms. Sankranti Devi, Research Scholar, Guwahati University, and Ms. Sangeeta Das, Research Scholar, Guwahati University, and members of the organizing committee, Dr. Jyotishmita Das Ma'am, Dr. Sandeep Chaudhary Sir, Dr. Smorajit Oja Sir, Rajarshi Deka, Juganta Jyoti Das, Hirok Jyoti Kalita, Keshav Jyoti Bora, and all the research scholar of the RS Forum, Department of Zoology, Guwahati University, for their constant hard work in making this webinar a grand success. Finally, I would like to thank all the invitees and participants from various organizations and institutions for attending this webinar. Thank you so much for your cordial cooperation. My sincere apologies if I have missed out any names. Once again, I thank you all. Thank you so much.
Thank you. Thank you so much, Priyanka Kashyap, for such a beautiful vote of thanks. And now moving on to the end of this program, I would like to request our professor and head Department of Zoology, Guwahati University, Professor Jugen Chandra Kalita sir, to kindly declare the closing of the webinar.
Yeah.
Over to you, sir.
Thank you, Sankranti. Thank you, Priyanka Kaisap, for your very nice offering vote of thanks to everyone. You name
Thank you,
all
sir.
the entities, our resource person, as well as our guests. I am very happy that Professor Viloljuti Sinha was the key person. He connected Ija Adriana from Malaysia. When I knew about her, I told Viloljuti Sinha immediately, please connect her so that she can speak. And today she spoke. It was wonderful. And also, when I knew about Sristilekha, you see, Sristilekha, a student, very meritorious from the beginning, from our school days, as Professor Viloljuti Sinha, head of the Department of Zoology, Borland University, mentioned categorically that from vernacular medium, he was doing so good. And now you see, she's getting a scholarship to study for her PhD degree for about 5 years in a very rich country, developed country, top-ranking university in Japan. So I congratulate both the scholars. I express gratefulness from Guwahati University to both the guests, okay, our Professor Hilojyoti Sinha and Professor Madam Yasmin, also talented personalities. We are blessed by their gracious presence today. I am so happy that the Girls College, which is Nagaon Girls College, again, ma'am was talking about girls empowerment. We encourage the girls. And ma'am, we connected that particular college where Dr. Sandeep Chaudhary, Dr. Smaraji Tuja, they work together. And from there, we are getting lots of helps. As Priyanka Kaisab already mentioned, Nagaon College Autonomous, Dr. Jyotismita Das, always with us. So dynamic this team, Sankranti Devi, Sangita Das, our Rajasri Deka, Keshav Jyoti, Juganta Jyoti, Virab Jyoti, and so many Jyotis with us. So therefore, we are proud of all of you. I cannot name each and every one. There are every possibility of miss the name because it's a huge group, it's a huge, huge group. And they are always together and they are
Vijay is also there, sir.
yeah. So they're very talented personalities. So I am really thankful to all of you. Our aim was to finish by 7 o'clock. It is 1 minute to 7. I wish all of you a very good time, future, and during this COVID pandemic. Please stay blessed. Take care. Thank you so much. Thank you
Okay,
all.
thank you, sir. Thank you so
Yeah,
much.
wonderful hosting, wonderful presentation, and wonderful organizing capacity of the team. This team is wonderful team. I finally thank the college, Girls College, Nagaon Girls College, Nagaon College, Berlin University, Guwahati University, and also the Patna Women College. Because, ma'am, this is the beginning only. You will have to deliver good language. When you spoke, I was observing. You are a good speaker, good
Oh my
worker, and your heart is so good. You talk about development of women. Thank you so much, ma'am. We'll meet again. Thanks all participant friends. You are without you, we cannot do this. So participant friends,
Thank
thank
you,
you so
sir.
much.
I also thank you for accommodating me in your webinar, in your program. You have a big heart.
Thank, thank you. This is the
Thank you.
beginning.
Thank
I
you so
invite you
much.
to talk. Yeah. Okay,
Thank,
Sankanti.
thank you so much, ma'am, for being there.
Okay. So this is the end of the session. It's exactly 7. We are on time, started on time, and also ending on time. Kristilekha, Ija Adriana, again, congratulations to both of you.
Thank you, sir. Without you, it was
Uh,
impossible. Thank you for giving us the opportunity.